Hi, everybody. Good evening. I will start sharing my screen then. You should be able to see my screen now. And okay, good evening. Welcome to the sixth live session for the NPTEL course, Cell Culture Technologies. I'm Pallavi. I'm a PhD student at IIT Delhi, and I am the TA for this course. So, just some basic things that we just go through every time. This is an 8-week course, and we've been through 5 weeks already. We have had 5 live problem solving sessions. This is the sixth one, 2 more to go. And as always, these Zoom sessions are being recorded. The recording is being uploaded to YouTube right after the session, and anything we discuss in this session, so primarily this PPT, that would go on to this Google Drive, and you will have access to all of this through the course page. Just scroll down, and there's a live problem solving session tab at the bottom. So you'll access all of this from there. There's also a discussion forum or a Q&A session, Q&A section in the course page itself, which you can use to ask any questions other than this live session. So we have 3 of you joining us. So I think some of you are joining us for the first time here. So would any of you like to introduce yourselves? I know a couple of you have already been here, but if any of you want to introduce yourselves, feel free to unmute yourself at any time to ask questions if you have any suggestions, any confusions, or you can use the chat feature to type in your questions or anything. It's a very small group, as you can see, so it's absolutely okay to interrupt in between. Not really an interruption, right? So just make it as interactive as you want. Otherwise, I'll wait for a few seconds and then I'll move on to the next thing. So if there's anything you want to share, um, and are you all done with your, if those of you who are enrolled in any course at the moment, are you done with your mid-semester exams for the current semester? Do you have a break at this time? Because I was expecting maybe in some class nobody turns up, so that would be when your mid-semester exams would be going on. But we've had attendance so far in all of these sessions. That's nice. So anything, anybody at all? Or if not, then we can start the session. So I do see some new names today. So to all of those, welcome. Um, and before we move on to discussing some things from week 6, is there anything you would like to tell us about? Would you like to introduce yourself? Would you like to ask something? Anything at all? I'll just wait for a few seconds and then we move on. Right. All right. So if there's nothing else, but again, feel free to unmute yourselves at any time or write your questions or anything in this chat box. In the meantime, I will start with what was planned for this week. So, so before we go, I'll just give a brief, quick 1-minute recap of everything that we have covered so far. So the first 4 weeks were more theoretical in the sense that we were not talking about actual experimental setup and protocols. We talked about what all things do you see in a cell culture lab, what all things you need to consider when setting up a place, and what are the basic aspects of cell culture. So primarily there are these things: the media, the atmospheric conditions, so maybe the humidity, CO2, and all of that, cells, the substrate. That's what we've been talking about for the first 4 weeks. And then in week 5, we've jumped into one type of a cell culture, which was fetal or embryonic neurons. And then, so at that time, there was this case study of a neurodegenerative disorder with the example of Alzheimer's. And we did go on and talk about that culturing embryonic cells would be simpler because they divide. It's easy to access relatively. So the dissociation, all of that would be slightly simpler. And since they divide, they're easy to maintain. That's the main thing. Because if you start with 10,000 cells, you will go on to 20,000, maybe 18, maybe 22, things like that. But for an adult neuron culture, which are non-dividing, you start with 10 and you just have 10, right? Or you lose some also. So it's just more difficult to work with them. So anyway, so that's where we were until last week. And now we dive a little more into neuronal cultures. So before we go on to what was discussed this week, I'll give you a brief, quick crash course in neuroscience. Again, this is covering a very, very small aspect of the nervous system, but I think it is relevant for what we are discussing this week. And also it's just a good idea to have to know this. If any of you are already studying neuroscience, so I do give this sort of a disclaimer before some of my discussions. So I'm not a neuroscientist by any means. I'm not studying a lot of neuroscience basics. So I'm explaining you based on what I know. I've tried my best not to make any mistakes, but in case if you are able to pick on any mistake, just let me know, right? But hopefully I'll tell you the right thing. Okay. So basically the nervous system, which is neurons and all of those things, we can basically divide them into two parts, the central nervous system and the peripheral nervous system. And as you can see in this diagram here, so everything that's labeled in pink, which is primarily just the brain and the spinal cord, that's the central nervous system or the CNS, and everything else, all other neurons. So when we say all other neurons, so you have broadly, we can classify all other neurons into two types. The amongst the yellow ones. So there are neurons which are taking up signals from your body to the brain. So for example, touch, heat, all of that. These are sensory neurons, right? And the second type is motor neuron, which is responding to any of the inputs. So your movement is being controlled somewhere by some neurons, right? So those are motor neurons. So broadly, there are two types of neurons in the peripheral nervous system, and you have a lot more types of neurons in the central nervous system. And central nervous system, again, depending on what sort of a resolution you want to look at, there are a lot of sections and subsections that can be done of the brain. So you have areas based on whether they are towards the outside of the brain or the inside of the brain, what particular function do they do, what particular sort of response or analysis they are responsible for. For example, for uh, for emotions, there's something else. For speech, there is something else, and so on. Right? Again, this is way out of the scope of this course, but you should know that there are different regions of the brain which are important for different aspects, and they have their own sort of neurons and non-neuronal cells and their functions. So last time, if you remember, we talked about the hippocampus, which, as you can see, it's not there in this diagram, but it's there somewhere in the inside in the middle of the brain. And we saw a small dissection video also. Again, for any of you who have not joined us last time, it's just a YouTube video that I've shared. You can go on to the slides for the last time, for the last class, and then just look at it if you're interested. But yeah, okay, so that's that. And then specifically talking about the cortex, which is often one of the most studied regions of the brain. Neurons are arranged in several different layers. And again, different functions, different age of development and all of that. Again, this is just for your information. What is relevant for this week's discussion is something about the spinal cord. So out of the CNS, so we broadly talking about CNS and PNS neurons. For PNS, we'll come in some time. But for the central nervous system, we talked about the brain briefly in the last week. So that's done. We talk about the hippocampus briefly and That's our region from the brain that we're talking about. About the spinal cord from the central nervous system, there's one diagram that I would like to talk to you about. So spinal cord runs down the back, right? So here, if you take a cross section of the spinal cord, you'll see a structure that looks like this. Okay, it's not all the same. It's not uniform all around. It's this horn sort of a shapes, and we'll come to this in a second. So, like I said, there are two types of nerves or neurons. So nerves are mainly bunches of neurons. All neurons together taking in signal, giving out signal, just so that there's a lot of it. So we'll use nerve and neuron interchangeably. So there are two types of neurons. One is bringing in the input to the central nervous system and telling it that, oh, I found some touch or some pain or some heat. And then there are the other type, which are some process happened in the brain or the spinal cord, then they're taking back the signals and telling the other part of your body to move, some sort of a movement. So motor. So I'll just zoom into this diagram. So you can see there's this thing that's coming in. So if you follow this arrow on both sides, there's this arrow that's coming in towards the back. So the direction here, there are two. So direction is, if I talk about the front and the back, so back is dorsal and front is ventral. Okay. So if you have this spinal cord, so this front part is actually the front of the spinal cord, which is like, if this is my spinal cord, then this part is ventral and this behind part is dorsal. So again, so this blue part that you see here, this is the back. So that's the dorsal bit. And this is in the front. So that's the ventral. Right? Ventral half, dorsal half. So the input, which is also called afferent, I've just put up these things here. Again, you don't necessarily need to remember all of this for this week. But what we discuss next, so all of this would just help you understand that maybe 1% better. That's all. Okay. So the sensory input comes in through the dorsal part. Okay. So dorsal is sensory. That's the input. It's called also the afferent neurons and all of that. Something gets processed. And then from the ventral part, things go out, which is your motor or the afferent or the output. Okay. So that's one basic thing. Now, remember, we talked about that a neuron is not it's not uniform all around. There is a central part which is called the cell body, which has the nucleus, and then there's a big axon, and then there are a lot of dendrites. Okay. So now, if for all of those neurons, if the cell body of all neurons which are doing the same task, if that cell body is at one particular space, you can assume. So let me just draw this out. Yeah. So this is sort of what your neuron looks like. Okay, terrible drawing. But if this is the central part, this is the axon, these are all the dendrites, so on, the cell body, all the dendrites. So if all of the cell body, okay, this is the cell body. So if the cell body of all the neurons are concentrated at one place, you can see that it's going to be a bulkier bit. Okay, because this is what's taking up the most space. The axon is relatively thinner, dendrites are even thinner. So the place where the cell body of the incoming sensory neurons, that is concentrated at some place. It doesn't happen for the motor neurons. It's just important to collect all the signal in one place before it goes in. So that's why the sensory neurons, the input neurons, the cell body for all of those is concentrated at one place on both sides. You can see this. So again, this is probably the right side, right? And this is going to be the left side. So all of those things, they are, so yeah, so all of that is concentrated in one place, and that just gives you a bulkier bit. So that is what is called I'm not, yeah. So this is the, you can see, so it's at the dorsal side, it's at the back side. So that is what is called the dorsal root ganglion. Okay, DRG. And this is what we will be talking about next. So this is one place where there are a lot of neurons. Just remember this. Dorsal root ganglion, dorsal side. Dorsal side is where you get the input from. And root is, so this is the cell body concentrated place. Okay. Again, let me know if there's any confusion at any time, right? Otherwise, if there's no confusion, keep giving me thumbs ups here and there, and I'll know that you're following through. So, anything else that we need to talk about here? So we'll just, so, okay. And then within the spinal cord, you see the structure. Again, there's a dorsal part and a ventral part. And these spikier structures, which are these like pointed areas that you can see on both left and right side, these are called horns. So dorsal horn and ventral horn, both. Okay. So, and yeah, so nerves are just bundles of neurons. So if you don't read the bottom part, the spinal cord, so there are neurons here inside, and then there are neurons that are coming to and from, right? These things. Can you tell me which is a part of the CNS and which is a part of the PNS? Any idea? What do you think? And it's okay if it's wrong, no problem. So the question is, you see there are neurons outside the spinal cord and there are neurons in the spinal cord. So which of these are a part of the CNS and which of these are a part of the PNS? They're all connected to each other through synapses. But which of these is a part of what? So neurons which are within the central and within the ventral and dorsal horn, so that is entirely inside the spinal cord, they are a part of the CNS. And then these root ganglions that we talked about, since they're coming from the body, they are a part of the peripheral nervous system. Okay, so just remember. So today we'll be talking about both of these things. Neurons which are coming from the dorsal root ganglion, we'll talk about them as a part of the PNS, peripheral nervous system, and cells which you extract from the ventral horn, which is this front part, that's going to be a part of the CNS. All right. Again, we'll revisit this once we come to that topic. And then also we'll talk about a few more cell types. So the first thing, of course, abundant neurons in both PNS and CNS. And neurons are the basis of the brain functioning, body functioning. So they are definitely one of the basic, most fundamental cells. And so they come first during development. Okay. So remember we talked about the E14 time point for a mouse or a rat, rat's gestational period. So the gestational period or the pregnancy goes on for about 21 to 23 days in these species. And by around E14 is when you can isolate. So for example, last week we had seen the example of the hippocampus. So by E14, you can isolate the hippocampus and dissociate it to get neurons, embryonic neurons, and you'll get a lot of neurons from that. But the embryonic stage may not be enough to get some of the other cell types. Now, what are the other cell types? So there are some cells which the neurons need. So neurons are, when we say that neurons are firing these action potentials, again, we're not exactly going into all of that in this course, but neurons are communicating with each other and they're like super fast signals which are going on. So what happens at the intersection of the neurons is one of the neurons is releasing some small molecules, some signaling molecules, and the next neuron is taking those up. These are called neurotransmitters. And then it's propagating that signal ahead. So there is, if this is the end of the input of one neuron, this is the start of, end of that output from one neuron, and this is the start. So somewhere in between, there is some empty space. Now, within that empty space, you'll have these neurotransmitters floating around for some time, right? So if it releases 10 molecules, it may take up the next molecule, the next cell may take up 8 molecules. So there's still something floating around, and there are a lot more things happening. So this extracellular space, so as to say, this does need to be cleared up, right? So plus there are a few more functions of support that need to be done, that are required within both central and peripheral nervous system. So that is performed by astrocytes or satellite cells. Astrocytes in the central nervous system, satellite cells in the peripheral nervous system. Somewhere you may also see astrocytes being written as astroglia. So all the non-neuronal cells broadly are called glial cells, where astrocytes are one of the most common ones. But yeah, so astrocytes, astroglia, they're the same thing. I'll come to the second point in a bit. Then there's microglia. So microglia, as such, it's not, maybe I should not classify them as glial cells. They're more of the immune origin. So they are there to clear up dead cells, not to clear up these neurotransmitters, but microglia have in general cleaning the dead cells or chopping up processes. So again, we'll not go into too much of details, but neurons, especially when they are growing, not at the adult stage, but when they are growing, they may extend their axon and they may like, okay, I don't like it here, so I'll chop it down back. So those plus minus events also release some extra things that need to be cleared up. So all of that task is with the microglia. And then we have ependymal cells. So again, we're not going into that today at all, but there is something known as a blood-brain barrier. So there is not a free flow of blood throughout. So between the blood vessels and the brain, there is an extra layer of cells, which is known as the ependymal cells. Again, the main reason why it's there is because you don't want a lot of, so blood is a good source, is a very nice source of sending anything anywhere, right? But that also causes that if you have any infection going on in the blood, it can go anywhere, right? That's the basis of sepsis also. So once the blood is infected, it could go anywhere. So just building on that, you don't want the brain. So brain is very, very important. So the body doesn't want the brain to be affected anyway. So the blood-brain barrier exists, which is mainly by these ependymal cells. And yeah, all of these have different sizes. They are formed at different time points, but mainly all of these come up after neurons. So when you're talking about an embryonic tissue, you may just find neurons. But when we talk about adult tissue, as we will in today's discussion, in week 6, that's what we discussed, that if you want to study the other cell types, maybe looking at the embryonic embryonic brain may not be your best bet. But even if you want to look at the embryonic tissue, it needs to be really late. All right. Now coming to oligodendrocytes and Schwartz cells. So as was explained in the lectures, if you remember, so this nerve conduction or electrical impulse, all of these are just related terms. So conduction needs to go on perfectly. You don't want any losses anywhere, as you do with wires at home, right? You don't want you want the outside of the wire to be insulated so that there's no signal loss, right? If you remember all of these optic cables, they are sealed in multiple layers so that there's absolutely no loss of signal, not just so that the other person doesn't get shocked. We're not just talking about electrical wires, but also any sort of signal transduction. You don't want there to be losses of any form. So there has to be some sort of an insulation. In case of the nervous system, the insulation is by something called the myelin, MYELIN. I don't have that written here. So this is called the myelin. And since it's in the form of a sheet, um, another sort of synonym for sheet is actually sheath. So it's called the myelin sheath. It's the right term. Now, the way the myelin sheath is wrapped around on the neurons of the central and the peripheral nervous system, that's one of the major differences in these two parts of the nervous system. So in the CNS, you have cells known as oligodendrocytes, and they have multiple processes. They look like they have multiple processes coming out of their cell body, and each of these processes can wrap around a part of a neuron, and they can wrap around the same neuron multiple times, and they can also wrap around multiple neurons multiple times. Okay, so you can see there's one oligodendrocyte, there are two wrappings on this neuron, and there are also two wrappings on some other neuron. So these are relatively bigger cells. On the other hand, in the peripheral nervous system, there are smaller cells which are called Schwann cells. And what they do is that just a single cell or just a group of single cell, they themselves just wrap around a part of the neuron. So again, there also you'll have multiple cells wrapping around the length of the axon of a neuron. But yeah, so it's just two different types of cells, two different ways. So Schwann cells don't have processes, they themselves get wrapped around. And oligodendrocytes, they wrap around using their processes. Processes means this extra projection. That's what we mean by a process. Okay, so far so good. Can I just get a thumbs up or an okay from any of you? Yeah, okay, all good then. Thank you. So now we come up to, so the main idea, so the last time we talked about a neurodegenerative disease. Now we talk about a disease where this function is lost. So we want an in vitro setup, and in vitro means we want a cell culture setup in the lab. If you want to study a disease or if you want to test drugs against a disease, where demyelination happens. So demyelination, as the name suggests, the myelin is gone or it's going, it's getting degraded. So these are called demyelinating diseases. And there are two more common ones that you might have heard of. So one is the Guillain-Barré syndrome, which affects the peripheral nervous system, and multiple sclerosis, which affects the central nervous system. Okay, so if you want to study diseases like this, what should be your setup? So the first thing is, so again, let's start with testing the drugs. So if you want to test drugs which can reduce the rate of this degradation or which can ensure that myelination happens better, if you want to study such drugs, you want to have cells, you want to have neurons, and you want to have, let's say, oligodendrocytes or Schwann cells, whichever of the myelinating cells, you want both of them. And then you test a drug and then you see if there's any improvement. Or if you want to study a disease, then also you want to have neurons, maybe. So in that case, you want oligodendrocytes or Schwann cells. Maybe they come from the patients. And then, of course, you want neurons. And okay, the neurons are fine. But then what exactly is going wrong? Are the cells dying? Are they losing their efficiency to wrap around? Are they losing their efficiency to secrete something? What exactly is happening? Okay, so in both cases, you want both of these cells. So now we come to where to get these cells from. So this is just a summary of what was covered in a lot of, probably like 2 lectures of this week. So the first thing is you want neurons, and the second thing is you want these myelinating cells. Okay, so amongst the neurons, let's restrict our discussion to embryonic cells. So there exists this mix and match that we can do. So you can take embryonic cells and then, embryonic neurons, and then for the myelinating cells, the thing is that as we just talked about, the embryonic cells or the embryonic brain, or whether or any other thing, the embryonic or the embryo does not have sufficient myelinating cells. So at the embryonic stage, there is not enough myelination to begin with. So that's how it's going across development. So typically, you would isolate these from postnatal organism. Postnatal means post-birth. So ideally, postnatal means they could be just like a little pup, or it could be an adult. But postnatal typically indicates younger age, but postnatal could be anything. So for neurons, we'll mostly be talking about embryonic neuron. So you're extracting it from the embryo. And the myelinating cells, we talk about you're extracting that from the postnatal organisms. Okay. So now in both of these cases, you can have neurons which are from the CNS, you can have neurons from the peripheral nervous system. So for the CNS, you could have so you could take any part of the brain, any part of the spinal cord, depending on what. So like we said, for in some diseases, you may just want to focus on one part of the brain, right? We saw that there's all of these parts of the brain. So if there's a disease you want to study, which only affects the cerebellum, for example, then you only take out neurons from the cerebellum. If there's a disease which is affecting only the thalamus, just take out the thalamus, and so on and so forth. Like last time we saw the example of the hippocampal neurons, right? So they were pyramidal neurons, there's a subtype of neuron, right? Or what you can do is you can isolate, or you can do from the spinal cord, you can have these motor neurons. So remember the ventral part, the front part that's coming from the spinal cord. So we'll call them to be a part of the spinal cord motor neurons. So you can have those coming out. So we'll talk about those briefly. I mean, that's the example that was discussed this week. And then from the peripheral nervous system, so CNS generally always will have a lot of neurons. They're abundant in neurons. So whichever part you take, okay, obviously there are glial cells, but whichever part you take, you will have neurons there. But in the peripheral nervous system, there are neurons, but there are no particular space, or there are not many spaces where neurons are abundant. You don't want to dissect the whole organism and just be able to get a small number of cells. You want to dissect a particular part where you can get enough number of neurons. So that similar sort of a region in the PNS, which has a lot of neurons, is the dorsal root ganglion, which we saw here. So the input that's coming from your body to the spinal cord. So the place where you see this bulge, that is where all the cell bodies of these neurons are concentrated. So obviously there are all of these neurons there. Okay, so this whole set is what you can take. All right. This is a small video that you can refer to, so which does which it's not the dissection, but it's just explaining you what the dorsal root ganglion is. It's a small 2-minute video, good video. I'll definitely recommend you watch this. So that's the neurons. And now we want to talk about the oligodendrocytes and Schwann cells. So we talk about postnatal organisms. So for the central nervous system, you can have oligodendrocytes that can be from your hippocampus, right? So when you're talking about the pyramidal neurons, you can take it from just there itself, so that the neuron and the oligodendrocyte, they are already belonging to the same space. When you're taking something from the spinal cord, then again, you can use the ventral horn to get your oligodendrocytes from the spinal cord, right? The ventral horn, remember this inside part? So that's a part of the central nervous system. So you can take that when you're talking about oligodendrocytes for the CNS. And then now if you want to talk about myelinating cells of the peripheral nervous system, we want Schwann cells. And now for the periphery, you will have again your dorsal root ganglion. So since there are neurons, there are obviously going to be myelinating cells there. And we want them from postnatal organisms. So the best, again, the optimized bit that people use, that's from very early rat pups. So like we talked, E1, E2 until E21, E22, that is when the uh, fetus is not born. That is pre-birth, prenatal, embryonic slash fetal. And now the day of the birth is called P0. And then each day after, so P again stands for postnatal. So P1 is the next day after birth and so on. So very early, young pups are what they are used to extract Schwann cells from the dorsal ganglia. Okay. So far, so that's the two types of cells we are talking about. Now comes the complexity. Oops, sorry. So last week, we said that when we are like isolating neurons from um, the embryonic hippocampus, there's not a lot of other cells that you need to worry about. Plus, when you're only interested in growing neurons, you can grow them in a setup which is only conducive to grow neurons in. So your substrate, well, substrate is not as big a problem here as the medium. So each type of cell needs its own growth factors to grow, simply to grow, divide, whatever you want to call that. So there's growth can mean both things. Making these extra processes also, making these connections and just surviving. And growing also indicates that they're dividing. So whichever of the two things the cells can do, for the cells to do that, they need an appropriate medium. Okay, we talked a little bit about neurobasal A medium the last time. There were a few additional factors like glutamax and what else did we talk about? I think B27, right? So all of these extra things can mostly they support the growth of just neurons. So that's fine. Now we have two problems at hand. One is when we are isolating an adult tissue or let's say a postnatal tissue, whether that's the dorsal ganglion or the hippocampus, now you have both types of cells. So you have an abundant number of neurons also, you have an abundant number of glial cells also. And when we say glial cells, you have, a lot of different types of cells. Okay. So your question, and so now your question is, do you just want to pick one of these types of cells, one of these cells that are there? Okay. Second, first question is you want to pick that. You just want to extract that. Second thing, now we will go into growing two types of cells together, right? We want to grow the neuron and one of these types of myelinating cell, we want to grow that together. So there are these two parts to the next part of our discussion is these two things. One, when you're isolating your tissue, now you have two or three different types of cells with you. How do you separate them into their different kinds? So there are two methods of separating cells. One is called density gradient centrifugation. So what you'll do in that is you'll take a tube and you'll have, again, this was discussed quite extensively. So whatever be the shape of the tube, you'll have different liquids of different densities. Now I would like to ask you, if I compare D4 and D1, which of them has a greater density? This is not a question from bio, this is more chemistry or physics. It's just about density, so probably more physics. So which of these mediums is the most dense? Which is denser? Which medium is denser? D1, D4? You're all able to hear me, right? Come on, this is easy. Which medium is denser? The one that's at the top or the one that's at the bottom? No, nobody. Come on, give it a guess. can just put it in the chat if it's not possible to unmute yourselves. No? Okay. Never mind. I'll move ahead then. So something that's denser gets settled at the bottom. Okay. So yeah, let me just do this. So this is the right thing. So this is dense. So for example, when you talk about water and oil, if you add both of them in the same jar or in the same glass, you'll see water stays at the bottom. Oil, you'll have oil on the top because oil density is less. So what you do is you'll have, let's say, 1 ml to 2 ml or whatever the range is. If you're talking about in a small Eppendorf, which is just 1 ml in size, 1 ml in capacity, then maybe what you're talking about is just 100 microliters each. If you're talking about a bigger Falcon tube, which is 15 ml or 50 ml, then maybe you're talking about a few mLs each. Okay. So different mediums, they could be different concentrations of sucrose, different concentrations of some defined chemicals like OptiPrep. So again, what you do is you'll have different concentrations put, and then on top of it, you're going to add your cell suspension, the single cell suspension. So the first bit stays the same. You isolate your tissue of interest, and then you do a single cell dissociation by whatever means, and then you get single cells. Cell contact is broken. Now what you want to do is you'll have that cell suspension has multiple cells of multiple types. Let's say you have 100 neurons, let's say you have 50 astrocytes, 20 oligodendrocytes, and maybe 10, what's remaining? Microglia, right? So what's going to happen is now, and then what you do is, so if you're aware of the principle of centrifugation, where you will rotate a lot of all of these liquids at different, at whatever particular speed. So what happens, depending on their densities, they're going to settle. So if it had just been water or just some water-based medium, then they would all settle to the bottom because cells are heavy, right? Cells will settle. But now what's going to happen, instead of water, you are using something which is denser. So sucrose is mainly sugar solution, right? So it's thicker, right? If you make like chashni, sugar solution, again, that's a lot of process. But since you're adding a solute, the density of that solution increases. So the highest density, highest concentration of sucrose goes here, then lower, lower, and lower. So cells are going to settle after you've centrifuged them, maybe 300 RPM, 300 revolutions per minute. After you've centrifuged them for, let's say, 2 minutes, 5 minutes at this speed, what you'll see is that you'll get different cells at different layers. So the main idea here is that, so cells, generally, if we assume that cells have a similar composition throughout, or I should say, they have a similar requirement throughout, and the insides are, it's like if you've spent the same resources in each cell, if you've sent 100 molecules into each cell, you have bigger cells and then smaller cells and then small cells, and then let's say you have these 4 different sizes of cells. So what you'll see is that the bigger cells, so let's say there are 4 things in each of these cells. Okay. So what you can see is that the smaller one is like super dense. Okay. So this goes here, and then goes the bigger cell, and then the bigger cell, and then the bigger cell. Okay. So anyway, so now you have these different cells, which you'll find in different layers. And then there are mechanisms, other more steps where you can just pick up cells from different layers, and then there you have it. All cells separated, and now you can add them into 4 different tubes. So you've got that. Now, Suppose I already give you a protocol that, okay, you use these 4 different concentrations of sucrose, do it for this time. The bottommost layer will have your astrocytes, the next layer will have microglia, next layer has um, oligodendrocytes, and the topmost layer has neurons. Let's say I tell you this already. Then that's fine. You already know what to do. But now you're working in the lab and you realize that, well, this is not doing a good job at isolating and separating out 2 types of neurons. Okay. So there are different types of neurons. And let's say the tissue that you're working with, let's say the hippocampus or in any other part, it has 2 different types of neurons. So now both of those so now both of those types of neurons are here. Now you want to separate these out. And you can separate them out based on densities that you know. But now you want to develop something more. So let's say you develop this OptiPrep, or you develop some new concentration, new gradient column material. So you will need to do some optimizations for that. So you'll say, okay, we want to introduce one more density. The other 3 densities look fine. So I'm not changing that. But I want to introduce maybe one more density. Maybe you're just adding another sucrose density. Okay, so you have one more now. And what sort of density difference are you talking about? So if it's so maybe are you changing just by a few grams per liter, or what sort of differences are you doing? So what is that new density going to be? And then fine. Okay, so now what you'll see, okay, you've got this extra layer here now. So uh, which of the 2 types of neurons, if you're able to separate them out, which neuron is at the top layer, which neuron is at the bottom of the 2 layers, right? So how do you confirm that? How do you confirm the identity of what you've got? So there are 2 things you can do. You definitely extracted that layer. You want to look at them under the microscope. So you mainly are what you're going to do is culture them, okay? Grow them independently without anything else. Grow them, see how they grow, look at their morphologies, and do some immunostaining. So if you know that, okay, these are 2 subtypes of neurons, let's say we call them subtype A and B, and they express particular uh, protein on their cell surface or within the cell or wherever. So then you can say that, okay, in my lower layer, I saw A. In my upper layer, I saw both A and B. So maybe the top layer is not like a perfect separation, because I could find both of these markers there. The other marker should not have been there, okay? But if you find A in one layer, B in the other layer, then you're good to go. So that's that. So again, this is like an optimization that would need to be done only when you're making something new, or when you're using sucrose for a new cell type, right? If let's say you are interested in another region of the brain where this protocol is not optimized. Suppose I give you the protocol for the hippocampus and you want to use it for the cerebellum, right? So in which layer do you see with cell, right? You would want to confirm that. You don't just want to go by me saying that, okay, since neurons from the hippocampus are there in layer 4, I would also see neurons from uh, cerebellum in layer 4. Maybe I also see some of them in layer 3. So layer 3 is now not pure. Okay. So this can happen. So these sort of optimizations and checks are very important, because as we discussed, oftentimes in cell culture, you'll be culturing cells for weeks and months. You want to be sure in the initial step that what cells you have are actually the cells you are looking for. Otherwise, 3 months later, when they finally grow and when you're actually doing your experiments, the actual experiments, you'll see, oh, well, it's not giving you the right result because it was not the right cell type to begin with. Okay. All right. Cool. Just give me 1 second. I'm getting a call. I'll just come. Sorry. All right. All right. So that was one thing. That is the gradient, uh, density gradient identification. Um, Actually, now I was just thinking about it. So maybe, oopsies, maybe this statement is not correct. Uh, because density has nothing to do with the cell type actually. I mean, it could be bigger cells if they have something like a vacuole or they have an empty space, they could be less dense. But I think I'll just remove this for now. It doesn't matter anyway. It's not going to affect our discussion. The concept still stays the same. The less dense cells, whether they're bigger in size or smaller in size, they could also be bigger cells, but whatever share the same density. So more dense bottom, less dense the top. All right, cool. Sorry for this interruption. The next thing is, so this is a density gradient identification. But again, what if two different types of cells, they have roughly the same densities? They may have minutely different densities, but not enough different that they can be separated in a gradient like this. So in that case, remember we just said that you are interested in two different cell types, which express two different markers. Now, the best thing would be if those two markers are expressed outside the cell. So when you're talking about a cell, you could have things which may be expressed on the cell surface and cell membrane. There could be things which are inside, right? So if something is inside the cell, well, that's not detectable from outside. But when something is outside, that makes it 1% better. We'll come to this in more detail in the next week where we discuss facts. But as long as you have a marker that can uniquely identify your cell of interest, you can isolate that, you can purify that with good enough confidence. It's a bonus if it's outside, but as we'll see, it's also possible when it's inside. Okay? But yeah, most, like the best case would be if it's outside. And we'll come to it why. So the only thing is that if it's outside, what you can do is, so I'll just briefly tell you what the antigen antibody concept is. So antibodies are they're generally denoted like this. It's called an antibody. We briefly saw some immunofluorescence images in the last week. We'll also look at some in this week just to explain some things better. So antibodies. So normally the way antibodies are generated, they're generated by the B cells in your body. Okay, B cells, which are the immune cells. So antibodies are generated against something. So this antibody, it can identify something. So let's say it identifies this molecule. So this molecule will be known as the antigen. Okay. So antibodies are very, very specific. They may identify the whole antigen or they may identify even just a part of the antigen. Okay. Again, we're not going into too much details of this. But let's say you were able to develop antibodies against this thing. So this part, this unique cell surface protein, and generally cell surface proteins are receptors, but whatever the cell protein is, let's say we are calling it the antigen. And if you have antibodies against this, then those antibodies can be used to select just this, because these cells are going to stick to those antibodies and you can just extract those cells. What we will be talking about next week, which is called FACS, which is called fluorescence activated cell sorting. Also, some places say fluorescence assisted cell sorting. I think it's the same thing, but fluorescence activated, that's what I've been using so far. So what you can do is, in addition to this antigen and this antibody, you can use a third component, which is fluorescence, some sort of fluorescence molecule. Now, what is happening is that this antibody is detecting this antigen and this fluorescence thing is detected this antigen. Generally, This is also an antibody, this fluorescence thing. It's just an extra thing. But anyway, so this fluorescence is tagged to this antibody. This antibody can attach to this antigen. And then what happens is now your cell just glows. Instead of sticking the cell to your antibody, we're not doing that here. So the first case is where you have this, your substrate, where now you have these antibodies attached. Okay, so that's the important bit. These are these AB antibodies. They are attached here. And then what's going to happen is your cells with these receptors, cells will stick. And then you can unstick them just like we did with your trypsin and papain thing, right? You're chopping off these extra connections. And then you've got your pure cell. All the other cells will not stick. In case of FACS, you're not sticking the antibody to any surface, but antibody has this extra attachment, which is glowing. Let's say it's red in color. And then this antibody, because these antibodies are small, right? They're proteins and your cells are like big. So the cells are this big, your antibodies are going to be this. So the antibodies will now just coat the cell, and now your cell will glow in that color. And that glow is the fluorescence glow. So then cells which glow green can be sorted, cells which glow yellow can be sorted. So each cell type can have a different color. But again, we'll talk about this in more detail in next week. So when you want to separate cells out, coming back, why did you want to separate cells out? Because in your postnatal organisms, there are very few sources or almost no sources where you'll just have one cell type. But if you're interested in isolating a particular cell type, whether that's your neuron or it's a non-neuronal cell, a myelinating cell, whichever particular cell type it is, to be able to isolate that particular cell, often called cell purification, cell separation, cell isolation, similar terms, you can use something like a gradient centrifugation, where you have a gradient column, each cell goes into its different zone. This can be great for some cells, it's much simpler, but it often lacks a resolution. In this case, as long as you have something unique on the cell, so we restrict it to cell surface receptor. Inside is also possible, but we'll come to that next week. If it's outside the cell, you can use this funda. Inside, you can't have your antibody penetrating into the cell, right? Cells often don't want anything extra going in. If you want something to go inside the cell, you'll have to temporarily chop up the membrane, which is a harsh treatment, but that's possible. So, but anyway, as long as it's on the surface, you can mark it and you can isolate it through either of these methods. All right. So far, so good. We are through 1 hour. I think we'll just finish up in another 30 minutes or so. All right. So now we have our two sources of cells. We have got neurons from embryo, either case, whether we are studying. So yeah, from the embryo, we have our neurons. From our dorsal root ganglion, when we're talking about the peripheral nervous system, we'll have our Schwann cells. So again, just a quick recap. You'll dissect out. So from the spinal cord also, you can have its neurons, whether you want neurons from, so yeah, so, oh, sorry, we're talking about the PNS here. So you want your DRG in this case. So spinal cord is dissected along with everything that's around it. And everywhere around is where you'll have your DRGs, your dorsal root ganglions. Whether you're talking about a mouse, that like embryonic rat or mice, or whether you're talking about the postnatal one. It was more, the rat was elaborated a little bit more, but in principle, this can work for any organism, right? So you collect your DRGs into whatever medium so that the cells stay happy. And then you dissociate your cells with trypsin or papain. Papain is gentler. And with trypsin, you have to ensure that you are not over chopping everything up. So there's often something known as a trypsin inhibitor that is used. So depending on how strongly associated or how much association there is between cells, Again, there are protocols which are optimized to tell you, should you add trypsin for 10 minutes? Should you add it for 3 minutes? Should you add it for 30 minutes? Okay. So that's there. And then if you're talking about an embryo, your dorsal root ganglion would only have neurons and they can divide. It's limited, but they can divide. And in case you're talking about the postnatal rat dorsal root ganglion, then once you've dissociated your cells, you need to do a cell separation. And we talked about the cell separation here. So once you've separated your cells, so amongst your cells, you'll have neurons. You'll have other cell types also, but we're primarily interested in either neurons or your Schwann cells. So what one setup can be used if you want to study the demyelinating disease of the PNS, which is the Guillain-Barré syndrome. So if you want to study that, what you can do is you can take embryonic neurons and you can take postnatal Schwann cells and mix them together. Now, when you want to mix them together, do you mix them both together? Do you add one versus do you add the other one? How much of each do you add? What substrate do you use? What should be the medium that you use? And as you can see here, neurons are not going to divide infinitely. Schwann cells, on the other hand, they are happy to divide as much. The thing is that in case of when it's inside the cell, when it's inside the tissue, when it's inside the organism, there's a lot of control that's happening, and we don't quite yet know what that control is. Okay, we know that there are things which individually both these cell types need to grow. But what are the things what sort of signals are there coming from the neurons, from the Schwann cells that are controlling how much of it needs to be wrapped around? How many do you want? Do you stop at some point of time? What happens? All of those things are not known in so much detail. So the best thing that people do is, again, all of this is just questions that you need to think about. A lot of discussion was not done in this week, and I'm also not aware of how all of this is done. But what one idea people use is you add your neurons, you add your Schwann cells, but then you also stop the division of Schwann cells at some point of time. And if you're starting with equal number, there's a very good chance your Schwann cells are going to outnumber the neurons. So since you're using dividing neurons anyway, you can let them grow for some time and then add Schwann cells, do it the other way, both of them together. In one case, as was discussed in the video, you can maybe put a layer of Schwann cells, allow them to grow, maybe stop the division, then add neurons. So there's no one right answer because normally in your actual division, neurons come first, then do the Schwann cells. But then we are not exactly controlling that also in so much resolution, right? But yeah, so neurons, then Schwann cells, again, relative amounts depend on what is the rate of their division and how soon or later do you want to stop that division. How do you stop the division of just Schwann cells? Because you don't want it, whatever it is you're doing to stop the division, it should not also affect any other process, okay? It should not affect the process of neurons making interactions. It should not affect the process of Schwann cells wrapping around. All of that is there. Again, it's a whole field in itself. I invite you to read about it. I'm also not 100% aware, so I will not be able to explain this any further. But of course, substrates, which we talked about, ornithine, so polyl ornithine and laminin, those substrate will support both of these. So substrate mostly stays the same. Media, of course, there are going to be additional things, not just your neurobasal and B27 and glutamax, but there would be a few other things which are important for Schwann cells to grow. So maybe there would be some more growth factors, HGF, EGF, there are all of these growth factors. Maybe there'll be a few more of those that you would need. But again, these protocols are always developing and that's there. Once you have this setup done, then depending on whether you used Schwann cells and neurons from a mice model which has GBS, or whether you're using a healthy one, so you can compare and then you can study. You can also test drugs like we talked about extensively in the last week's class, that you can add drugs and you can see if the myelination is better, if the myelination is poorer, how is it working. Is there a degradation that are you able to stop the degradation? Are you able to ensure there's more division? Are you able to ensure there's more secretion? All of that. And then we move on to the central nervous system neurons. So from the central nervous system, we'll talk about two cases. Again, the funda, now you know what you're doing. So in case of the hippocampus, again, you take out the neurons, you take out the hippocampus, dissociate, separate the cells, take whichever one you want, and then you're plating. If you want, instead of the brain, if you want to look at the spinal cord, then remember we saw this diagram with the first slide today. So what is at the back is dorsal, which is coming from the body, and then this is ventral, which is towards the body. So this ventral part is what is used to get your neurons and oligodendrocytes from the for the CNS, if you study, let's say, something like multiple sclerosis. All right, so that's that. And then, so yeah, so I think there's just one final slide that we'll talk about. This is, again, I've just added some images from some papers, just to explain a few things better. But before we go there, are there any questions up until this point? We'll probably be able to finish like soon. So, any questions so far? Is it too boring? You can give me some feedback. I'll keep this in mind. Yeah, no, okay, all good. Fine. Okay. So yeah. So now when we say that, okay, we've made these myelinating neurons, how do you, so one thing that we did not talk about is there are these gaps. Okay, it's not this insulation is not perfect. So these gaps are known as, can I just put it maybe a gap? So these gaps here, in both cases, you'll see there are these gaps, which are called nodes of Ranvier, nodes of Ranvier. So the thing is that you don't want the signal to go on along as one go. It goes on in waves. It goes on in like peaks. It goes on and then it dies down, and then there's another peak. So what happens at these nodes of Ranviers is there are sodium ion channels. So sodium ion channels are controlling the local concentration of sodium outside the cell. So in neurons, you will mostly see sodium and potassium ion channels, although there are also chlorine ion channels. But the main player, the main player is sodium, but also other things. So if I remember correctly, sodium is more on the outside of the cells. So you remember, you would have known when people are given these electrolytes, or when they're given drips. So that is mainly sodium, right? Some salt solution. So sodium is abundant in that. Sodium is abundant on the outside, and these sodium ion channels, they can regulate when the sodium needs to go in and when it needs to come out. So sodium can only go in. So there are these across the membrane of cells, there are different, so like we talked about membrane proteins, membrane receptors, there are also like proteins across the membrane which can control what goes in, what goes out. If something is there on the so for example, if this is your lipid bilayer, so right, so this is just this side, okay, I'm just talking about one of these sides. So this thing, so what you'll see is there's some some sort of a pore in that membrane, which can control. It's a controlled thing. It doesn't always stay open, doesn't always allow things. So, so this is inside your cell, and this is outside the cell. Okay. And then of course, you'll have another layer here. So this is inside, then again, this is outside. Okay. So now you have a lot of sodium ions here. Okay. And you have fewer sodium ions inside. So now sodium can go in. So think going across in the direction of high concentration to low concentration. So sodium can go from outside to inside when this channel is open. Okay. Now that is, so now this thing cannot happen when there's this insulation, but this can happen at these nodes of Ranvier. So nodes of Ranvier have a lot of sodium ion channels, which allow this trans movement. And this is important for the action potentials. Again, we're not going into too much details because it's not a neuroscience course, but it's a very simple concept. So you can definitely read about it. But now when you want to test if the myelination has worked or not, you want to test if there is myelin in the first place. How do you, so what you did was you added neurons and you added these myelinating cells. But what's ensuring that they're in contact? What's ensuring that this myelin is being generated, this sheath is being produced? So what you can do is you can tag. So if you just zoom into this image here. So let's just look at this first panel for now. So I actually edited out something from below, which was actually important. But anyway, so what you can see here in this case is, so there are these blue circles you see, that's the nucleus. Okay, mostly 99%. If there's a blue stain, and especially if the blue stain is circular, that's the nucleus. So that's DAPI or hoist. There are these two stains. And then you see this MAP2. So MAP2 is very similar to beta-3 tubulin, which we talked about last week. So these are going to stain your axons. Okay. So MAP2, which is microtubule associated protein 2. And you can see here it's in these lines. In all of these cases, it's in these lines, right? So that's your neuron. And then MBP, which is myelin basic protein. I should just write this here. So that's the myelin basic protein. And this MAP2 is, it's a neuron marker. Okay. Neuron. Yeah, right. So what you can see here is that there's a lot of myelin that's being produced, right? So it's there. And then in the second case, this was actually some sort of a disease model. So you can see there's not a lot of myelin being produced. And then they treated some disease, or they rescued that phenotype. And now you can again see that there's a lot of myelin that's being produced. So initially, this is the wild type, this is disease, and this is treatment. So maybe I can just write that here actually. I did not realize I've messed this up. So we can call this the wild type. This is your disease, and this is after treatment. Okay. All right. So that's one thing to check. So that myelin-based protein is being produced. Now, again, of course, if you want to test if the myelin basic protein is, so at this resolution, you probably can't see if the myelin protein is around your neurons or not. So you can go on and do these immunofluorescence. Again, these are called immunofluorescence because these were the same concept of FACS, what we just saw in a couple of slides back. It's the same concept now. So instead of this receptor, now it was myelin itself, or myelin basic protein. And now that's the antigen. Now you have the antibody against the MBP, and it's tagged with what color? Red color. Okay, so that's the same concept again. So you can zoom into this and then see it better. as you can see here. So what you also want to check is that are there enough nodes of Ranvier that are being there? Because you don't want it to be like a perfect insulation also. So against the node of Ranvier, like we saw that there could be, there are ion channels, sodium ion channels. You can have antibodies against that. There are other proteins also. For example, this thing, Casper. See, I think it's something associated protein. I'm forgetting what C stands for. Some C associated protein. And MAG also, if I remember correctly, that's also some sort of a myelin protein. So MAG here is telling you again. So this is again against myelin. This is again your node of Ranvier and neurofilament here. Again, that's also a neuron marker. So you see your MAP2, neurofilament, beta-3 tubulin, they're all neuron markers. One study is using one thing, the other study is using something else. That's absolutely okay. They're all great. So what you can see here is that, so this blue thing, the entire thing is neurofilament. And then here you see a lot of red. You don't see as much red here. There's a little bit, but you see mostly reds away from this green part, where there's this Casper, which is a node of Ranvier related protein. And then in the middle, you actually see that it's empty. So if I did not check if there's other images, but if you would have some antibody that's staining your sodium ion channels, then they would come up here. Okay, but you can see that very well there's some very particular empty space, empty as in which does not have myelin and has something else. So this is very, very localized to a small part. All right. So you've checked that MBP is being released. You're checking that there are nodes of Ranvier across your neurons. And then the main reason to have your uh, myelination is so that your cell, these synapse transmission electrical recordings, they are better. There's a better signal conduction. So there are two things that, so again, these are just complicated figures. What I would like you to look at here is that there's this unmyelinated type of neuron, some recordings from there, and then there's some other sort of myelination, less myelination, more myelination. Also the size of these neurons. So again, we put these two in one category and this as the other category. So what you can see here is that in case of the unmyelinated neurons, whatever sort of signal it is, so you can take the signal through the center, away from the center, whether it's like in like a particular node, it's being sent into, or if it's across the cell, whatever sort of recording there is, you can see that the signals are weaker. Okay, it's so you'll often see this sort of a characteristic graph. So you see this sort of a graph in both cases. Okay, in case of the large myelinated neurons, small myelinated neurons, you see this very fine up, low, up and down peak. But in case of the unmyelinated neuron, you don't see that. So what you can do is you can take recordings from, if you would have just cultured cells, just cultured neurons, and if you culture both neuron and your myelinating cells together, what sort of signal recordings do you get? That's one thing. The other thing is also the peak value, which is also evident from this graph. So you see the peak here. If this entire thing is like this is the scale for 3 microvolts, maybe this is 4 or 5 microvolts, and this is like definitely less than 3 microvolts, right? You can see the height here and the height here. And also, actually, if you see the x-axis in this case, this is a very sharp peak. So this x-axis, the scale that you see here, that's just 0.2 milliseconds. So maybe this entire thing is something around 5 or 6.5 or 0.6 milliseconds. But now in this case, what you see, The scale here is 0.4 ms, 1.4 milliseconds. So this entire thing is probably something around 10 or 12, like 1.2 or 1.5 milliseconds. You can see it's already a slow signal. So both the peak, the intensity or the amplitude, and the frequency. So how quick it is, how fast is the signal moving. The same thing, another paper, just some examples here. I would not be able to elaborate on what the two said here are, some uh, onset signal and the peak signal. But mainly if you just focus on this graph here, the two things. So if you just have neurons, and if you have neurons and Schwann cells both, and this is the number, 75,000 neurons, and if you have 75,000 neurons plus 25,000 Schwann cells, then the speed that you look at here, this is meter per second, so how fast it's going. So you can see that the speed is higher when you have, this is, oh wait, this is something else, right? It's the other way around. Oh yeah, okay. I think this is not what, it's not indicating what I wanted it to show. Forget this graph then, okay? I'll remove this before sending it to you. But what you can see from this, the first plot actually is, it's telling you both that the height, the intensity or the amplitude and the speed, it's both better when you have myelination. Yeah, maybe, I don't know what this is. It has to be some sort of velocity, but I'm not sure why the velocity is lower when you have Schwann cells. Okay, anyway, so that's that. And before, I think I just have like a handful, 3 or 4 quick practice questions, because a lot of it was already covered in our discussion. So we'll just quickly go on to it. I would really like if some of you could quickly send in your ABCDs here. So, oopsies. So which of these is not a cell of the nervous system? Neurons are there in the nervous system. Astroglia are there in the nervous system. Schwann cells are also there in the nervous system, but nephron, that's a part of the kidney. So nephron's not there. Just don't get confused between nephron, neuron. That's the thing. Astroglia, what are they? What are their functions? Astroglia, astrocytes. What are they doing? Now, so we talked about oligodendrocytes and Schwann cells, which form myelin. The immune cells of the brain are your microglia, and astroglia are to clear up any small molecules. So which is there to maintain the homeostasis. Homeostasis means that it's like a status quo, that everything is as it should be. No complications, no extra things, no less things. So it just ensures that you have enough of these sodium ions, and all of these ions and other neurotransmitters are cleared up from the synapse zone. Okay. And then what's the function of the microglia? So we just saw that microglia are the immune cells of the brain. And again, we saw this already. So density gradient centrifugation, it utilizes a column which has what density top to bottom? So remember we saw this here. So your density, so topmost has the lower density, so lowest density, and then top to bottom, there's a increasing density. Okay. Again, so yeah, so let me just quickly go through assignment 5 solutions. I really hope this open here. So in the meantime, I'll just log into my course page. So in the meantime, do you have any questions? So after this, we're left with 2 more weeks, 2 more classes. So have any of you registered for the exam? The exam's in the last week of October, is it? Lecture week 5 assignment solutions. Yeah, okay. Anyway, I'll just walk you through quickly through the assignment solutions. So primary cells are the cells which are obtained from animals. A defined system in cell culture, it has to be both chemically and physically uniform. So you know what it looks like, what are the compositions, temperature, pH, shape, size, amount, everything, right? So surface properties, and they should be repeatable also, of course. So we talked about mechanical dissociation and enzymatic dissociation. So why must air bubbles be avoided when you have a mechanical dissociation performed? So because they can damage the tissue. Papain's enzyme, which is there for enzymatic dissociation, that's obtained from papaya. A hemocytometer, we saw images of that last week. That's there for counting cells. It just has this grid and it's uniform. So it's like if you add 10 microliters cell suspension into it, if you're counting 5 cells on an average, then that's 5 into 10,000 cells per ml, per microliter. Sorry, no, per ml. 10,000, that into 10,000 per ml. Okay. But yeah, anyway, so hemocytometer is there to count the cells. For an adult hippocampal, for adult hippocampal neurons, which enzyme is preferred? So papain is there. Trypsin can also be used, but for neurons, trypsin can be a little bit more harsh because you don't want these processes to be destroyed in this dissociation in the process. Neurobasal medium is supplemented with B27 to grow. In this case, it's there for hippocampal neurons, but it's optimized for hippocampal and also other brain neurons. So neurons in general. And neurobasal is for embryonic. Neurobasal A is more for adult. Mechanical dissociation of the hippocampal tissue is done using. So it's pipettes. So what you're just doing is just you're pipetting up and down, but it needs to be gentle. It doesn't have to be harsh pipetting. In Alzheimer's disease drug testing, the synapse number can be visualized using what? So you can do fluorescent antibodies. We saw that if you can have a presynaptic molecule and a postsynaptic molecule together, let's say one is red in color, one is green in color. So wherever you have an overlap, which shows up as yellow, then you know that there is a synapse. There's an actual synapse that's forming. Because see, ends of all neurons would probably have that red color. The starts of all neurons would probably have that green color. But only when they interact, when they're close enough, will you get a yellow fluorescence. So random 2D neuronal cultures, which is what mostly is done, that is not accurate because they don't mimic the real brain region, right? So that's your assignment 5. And if I still have you on board, then that brings us to an end of today's session. So this is recorded. I'll upload it to YouTube. I'll upload this PPT. I think I'll just remove this because it's not telling you. Maybe the study was about something else. But yeah, anyway, the crux still remains that with myelination, your velocity and your amplitude. So the frequency reduces, velocity increases, amplitude is also higher. So better signal transduction. Okay, so that brings us to an end of today's session for week 6. The deadline for this week's assignment is day after tomorrow, 1st October, Wednesday. And I'll meet you again next week on Monday, which is 6th of October. The Zoom link stays the same. You'll get a reminder email on Sunday, and I will see you then. Again, if you have any questions now, I'll be here for another minute. And if there's anything other than the course also that you would just like to discuss, we have some time, so I'll be around. If not, if there's anything else you want to ask at any other point of time. So like I was mentioning, there's this Q&A option. You can just go in there, ask your questions, and I think both of you have been attending this class anyway. There's just one of you now. So the problem solving session links are here. All right, then that's about it. I will stop sharing my screen and also stop the recording.